We're going to do just a quick introduction of our speakers again as we set the stage for those that might have not have been here early in the morning. But to my left, immediate left, is Dr. Justin Buchanan with Simplot, and I think big Pokes fan, right, at Oklahoma State? Yep, did a 10-year at UC Davis and has been with Simplot for 7 years. Is that correct? Awesome. Okay. And then to his left is Dr. Brian Vander Ley at University of Nebraska-Lincoln, DVM. And I forgot to ask you this. So you were a native of South Dakota. Did you grow up a Jackrabbits fan? Yeah. Okay. They've had quite a bit of success lately. I think we've got a couple Jackrabbits fans in the audience. And then to his left is Dr. Scott Speidel, a native Californian. And I'm going to do an introduction as I also introduce Dr. Enns. I heard a rumor that Dr. Mark Enns and Dr. Scott Speidel were both roommates with our esteemed EVP Wade Schaefer at one point in time. Is that true?
I was the slumlord.
You were the slumlord?
Yes,
okay.
I was.
We might need to have a panel next year's Fall Focus to really dive into those stories, because I think that's a really beneficial thing to our audience. So, and then on the far side of the stage is Dr. Tim Holt. I think it's easy to say you are the PAP guy, but usually when you're PAPing, you're wearing a Broncos hat.
Absolutely.
So I'm just curious where that Broncos hat is
I
today.
have 7 of them in the truck.
Perfect. So we'll need that next time. Okay, so to kind of summarize where we're at, I think, Dr. Vander Ley said this best. I think one thing that all 5 of these PhDs can agree on is that we care about the health of cattle. This is something that is relevant to our industry, and we care about improving health. And like Jackie mentioned earlier, there are different ways to look at this topic. It's a complex trait. There's a lot that goes into it. And so we have 5 highly intelligent individuals trying to tackle the same issue from a lot of different angles to try to help you all make smarter decisions. And so I think we'll set the stage there. And I'm going to start off a question first. And so as those in the audience have questions, please raise your hand. But to get us started, I think I want to take it a little bit at a higher level. And Dr. Vander Ley, I want you to start, and then others can chime in. But give me just the perception of the feedlot industries. I guess their perception on congestive heart failure. What are the feeders in the beef industry? What do they think about congestive heart failure? Is there, what are their attitudes towards the genetic component of this? Just what are their perceptions?
I think the feeders that I interact with there's a couple of important themes. I think one theme that they're very concerned about is that it's an escalating problem. We had a number of feedlots that were involved in the discovery stuff that we did a number of years ago, and one of the things that was really interesting about that is we spent a lot of time both with owners, managers, and also with the feedlot employees. And one of the themes in the interactions, we spent many, many hours working side by side with those people as we collected samples and stuff like that. And one of the themes that really emerged from that was there was a point when most of those feedlots remember not having very significant problems. The first the first report of heart failure we can find in the literature relative to feedlot cattle, it's like the 1970s. There was some work that was done in Colorado that reported these heart failure cases that it was basically a frequency report. There wasn't a lot of investigation that we could find. And there was a little bit of extension work that we found in probably the early 90s in Nebraska. But most of them pointed to a period of time in the single-digit 2000s where this really raised to the level of awareness for them, where they're like, whoa, this is getting significant and troublesome and quite problematic and expensive. And we've done some of the frequency work that you've seen other people relate. And what we found very quickly was that feedlot producers really remember, for good reason, the cattle that tip over on their way to the truck. Those are very, very expensive cattle because of the investment that's been put into those already. But when we looked, we actually found it all over the place throughout the feeding period, which I think your data would support that as well. So I think the concern that characterizes a lot of the interactions with feedlot people is that it's escalating. And I think what makes them nervous about that is that they don't feel incredibly enabled to be able to directly deal with this problem, which is part of the reason we're sitting here talking about it today, because I think one thing that we can all agree on, regardless of approach, is there's a genetic element to this, and that is not in the domain of control of a feedlot. And they feel a little bit powerless to change something that's really hitting them hard.
Dr. Buchanan, you're directly involved in the industry. I mean, this is something that this is a perfect example of industry identifying a problem and investing, I'm sure, quite substantial amounts of dollars, time, man hours, personnel, to really try to solve a complex problem and come up with solutions. What do you think the responsibility of the industry is to trying to address some of that? And also, what does it mean for Simplot to be involved in that sort of solution solving?
That's a great point. We've invested a lot of money in this research project. We're very careful about where we invest, and this project's been one of the best outcomes that we've seen from a research project in recent memory. It's a big dollar amount, but when the outcome is dead cattle in the feed yard and they start piling up year after year after year, that justifies that investment. And our larger organization is behind us in figuring out what's going on with this trait. In terms of the larger industry, as Dr. Vander Ley mentioned, one other thing that we've noticed is that the accuracy of diagnosis is also increasing over time. 10, 15 years ago, those animals in the dead pile maybe wouldn't have been called a heart failure case. Today, there's been some great work out of Kansas State in helping the accuracy of the diagnosis, and that's also increasing over time. So we have to take that into consideration when we look at what's been the frequency as we go forward. More feedlots are becoming aware of this trait, and they're getting better at figuring out which of the deads are true heart failure cases.
So, Tim, I want you to try to elaborate a little bit more on the physiology. What is the physiology differences between low elevation PAP and high elevation PAP?
Yeah, it's a great question. So my thought is that if I'm comparing high elevation PAP to low elevation PAP, we got to go clear back to that diagram and go, what is the initiating cause of all this? And it can be feedlot, it could be altitude, it could be whatever. But we're going back to the part of that alveolar hypoxia, and then what that initiates. That initiates in human terms, it's called the hypoxic cascade. Same within humans as it is in cattle. So the difference is that when I'm at 10,000 feet papping or at 9,000 feet papping, then the odds game is that I am papping animals that are experiencing hypoxia because of elevation. If I go to Amarillo, Texas, and I pap, they are actually just exactly what everybody has said. We're not experiencing alveolar hypoxia, but we are experiencing hypoxia for some unknown reason. And that unknown reason goes on and on and on and on. And the reason and Justin said, maybe yes, maybe no about the same pathogenesis of it, meaning it's the same cause of it. But if I go to Amarillo, I have numerous cattle that are testing in the hundreds, right, up in the 90s, 80s, and it's still the same thing. Remember, the PAP test tells you, are they hypertensive or not? It doesn't matter where I'm at. If that is a high score, I have gone to Florida, to the Gators, and did some of their herd there, and I had a couple up in the 80s even in Florida. What does that mean? It means that they had actually probably a congenital defect. But the moral of the story is, what I'm checking for with PAP is to make sure this animal is hypertensive or is not. The etiology is still the same in some ways, is that hypoxic cascade was set up, and that's what's missing, because when you start looking at what causes all that to happen, it can be numerous things. And then the reason I admire Brian and Justin and those guys, when they look at the genetic aspect and us these guys putting the EPD together, there's so many contributing factors to that. There's how does the heart respond? That's genetic. There is what's the elasticity of the lung tissue? That's important. Thousands and thousands of things go into that end result of death. So the PAP is the same. The thing that concerns me, and it concerns me more and why I love sitting here, is when I look at an animal that died in the feedlot and I take them apart and I do histo on the heart, histopath on the lungs, I cannot tell the difference between one that died at 10,000 and one that died at 5,000 when I look at histopath.
So to Dr. Vander Ley and Dr. Buchanan, You mentioned that we're doing perhaps more necropsies. We're trying to diagnose on those dead cattle the specific cause of why they're dead. Are we noticing physiological differences in the heart change over time in that period, or how are we approaching some of that?
We certainly see it as this is a trait that develops through time. It kind of makes sense that a heart score 3, if we kept feeding that animal and he stayed alive longer, that might develop into a 5. Yeah, so there's certainly a continuum in the development of the trait. That's I think what part of confuses the genetic analysis of this trait is if you catch an animal and he died for some reason and his heart score was a 2, can we peel apart that animal had the genetic factors that were going to lead him to be a 5, or did we just catch him at the wrong time in the feedlot?
So what we do when we train feedlot necropsy technicians is we kind of give them this prioritized list of things to do. And what's happened is we've expanded that prioritized list to actually look at the heart more often. So that's become part of the standard. So I do think we see more heart pathology because we're looking for more of the heart pathology than we used to. And we often don't take those to the level that Dr. Holt described, where we actually do microscopic exams and look at histopath. There is some interesting work that's been published that Dr. Craftsher did this, where what we see in feedlot cattle is complete, like all of the cardiac muscle is affected. There's no elevation, and correct me if I'm wrong, but it's primarily a right-sided problem, and what we see in the feedlot has lots of chronic looking changes, which highlights the problem that Dr. Buchanan has pointed out, is that I don't think we really know when it starts. And because we only get to look at their ticker once, because when we get to look at it, they're not in possession of it anymore, we don't really know how the disease unfolds that well, and it creates some challenges. But I think we're accumulating more information because we are looking for it more and we find more of it.
Yeah, the timing of the disease is definitely a question, is what Brian talked about. We have another study Dr. Milt Thomas did that he worked in eastern Colorado with a herd that had some heart failures prior. And we put those in the feedlot, and Dr. Holt papped those. And when they came into the feedlot, we could have told you who was going to have problems. Okay, and referring back to Dr. Holt's slide presentation, the picture of the red calf you had there where they lost so many, that was prior to weaning, right?
Correct.
Okay, and we also have looked at, in Dr. Speidel's study, where we've done serial paps, and in the study where all of the pictures come from, I think we papped those calves 5 times, where we outline the heart score. The timing of that is a bit different, but the death all ends up at the same point for as far as chronic heart failure. And we are getting better at diagnosing this, and so there seems to be a genetic component at which this starts to occur, in my opinion, and I don't think we have that fully figured out. I do know that Dr. Speidel's study, if you think about his correlation between that last 3 to 4 weeks before harvest and heart failure, correlation was 0.94. So at that stage, right?
Yeah, genetic correlation.
Genetic correlation. At that stage, they're very highly related. I mean, that's even greater than our carcass ultrasound and carcass EPDs. That's a higher genetic correlation at that stage, that PAP then, and the heart score they're going to have 3 to 4 weeks later at harvest. I may have just muddied the water, but.
No, and I think we have a question from the audience. Callie, is that right?
On the feedlot situation, you indicated that at the end of the feeding period is when you start losing more cattle. If a guy shortens up the feeding days, I know economics comes in here. If he shortens up the feeding days, is that going to help? Then secondly, I'd like to know those cattle on the kill floor that's coming down, and the 4s and 5s, those hearts, are they correlated back to the carcass, and is there any problem with the carcass, or what do you do there?
I'll answer your 5. Every 5 I've seen come through the plant, and we have a lower incidence of 5 than Justin does. We've seen about 4 or 5, they're all condemned. Because when they open those cattle up, there's so much fluid they're off the rail to another path, the 5s, if they make it that long.
Yeah, you said it. It's the economic calculation of how many days do you feed them versus, what's the outcome from heart failure. We have ran this in our selection index. So if we try to create a model that takes into account heart failure, death, the 3 most important traits in that economic index are carcass weight, BRD, and heart failure, and they rank pretty similarly. Carcass weight being number 1. So yeah, it becomes a game of, do you treat this entire group differently and have a different economic outcome, or do you treat them the way we are today and you end up with the same rate of heart failure that also hits you economically? So it's kind of a balancing question in that regard.
I see 2 more questions.
In relation to Lundgren's work in the 1950s, he actually measured heart and lung weight compared to carcass weight. And we all know that carcass weight has increased 30-some%. And my question is has anybody looked at it from the anatomical angle as far as have we inadvertently selected for carcass weights and kind of missed the anatomy of the cardiopulmonary system, and that's what we have inadvertently got at this point. So I'd like to hear some comments, particularly from the magician over there.
Yeah, and add anything you want. One of the things that's going to just harass Brian just a little bit about is that when he started talking, he mentioned the 60s and early 70s, and he looked at me. I don't know what, So, but yeah, that's a great question. Because, yeah, we do. Every kind of heart that we harvest, not necessarily at the plant because we're not allowed to touch those, but if we harvest anything like in one of our studies in eastern Colorado or everything, we are weighing those hearts and we're measuring the weight and looking anatomically at them, and there has been no change in heart size. I have not seen dramatic changes in heart size and or lung size compared to carcass weight. Have you guys looked at that?
I think we haven't looked at it directly very much, but I think one thing I always try to think about is what would be the evidence that it's easier to disprove something than to prove it, because and the evidence that we have that may not be as big of a factor. We've actually looked into doing that, and we're trying to figure out a way to. It seems like a really easy thing to do, and then when you dig into it, it turns out not to be nearly as easy as you think. And we've really run aground. Even things like how much tissue do you take when you cut it off before you start weighing becomes troublesome. So the question then that we've asked in our group a little bit is, what's the evidence that might not be playing a role? And I think one significant piece of evidence that we still have is that we have millions of cattle that make it to harvest very successfully with some of the biggest carcass weights we've ever seen, and have still we still see a lot of normal hearts. So we have a lot of survivors despite the big carcasses and the fat that we get put into those animals. So it provides at least some evidence that relationship may not be super tight, or it's modified by something else.
I think we have a question over here.
I think the last beef quality audit, we had a beef quality audit in, I think, 2016, and the last one just recently came out. And if you look at what's happened to yield grade over time, we've significantly increased yield grades since 2016 to 2023. And I'm trying to think about what have we done in the industry that's contributing to this problem. So one of the obvious ones is fatness, so what I'm interested in is if any of you want to address the issue of what are the changes, and it kind of, this gentleman kind of asked the same thing, but what are the changes that we've really had that are really contributing to the problem that we're actually seeing?
And just ever so slightly, do you want to tackle that one?
This one or the
This
previous
one, or the,
I have
and
a
the previous
comment on
one.
The previous one too.
Comment on the previous one, and then also this one.
You know, that's a really good question, and it's something that I've thought about quite frequently. You know, if you look at I presented some information at BIF up in Calgary this year, and I had, you know, just lung size with respect to the species. So I had some inflated lungs from a horse versus beef cattle, and if you truly look at the size of the lungs with respect to the rest of the body, cattle are at quite a bit of a disadvantage in terms of their cardiopulmonary system when you compare them to other livestock species, you know, horses and that type of thing. So, you know, I always struggle with why, you know, how to explain why we are, you know, seeing some of these changes now, and you know, given coming back to your, you know, your original question on the changes in yield grade over the past however many, you know, since 2016, I think is what you said. The geneticist in me is going to say that if this is truly genetic, we're just starting to see some of these changes, because, you know, if you think about generation intervals and turning over generations, we're about at, you know, one generation away from where we were back in 2016. So maybe some of those yield grade increases that we're seeing now are more due to fat thickness and, you know, fat cover and everything else, and that's kind of how I'm justifying that in my head. But I also, you know, I've throw this out to everybody that I sort of talk to, you know, around the table after meetings, and I just wonder if you know, while we might not be actively, you know, kind of opposite of what you said, but are we not actively sort of putting some slight downward pressure on that growth, you know, if the cardiopulmonary system or that waste is not increasing, and that's might be explaining some of why we're seeing some of these increases now. So, you know, I've got no data to support it. It's just sort of as I sit there and, you know, drive and I'm thinking about things and questions pop into my head, but I have no real ways of answering them other than the fact that, you know, if you look at how carcasses have changed over the past, you know, number of years, you know, they're feeding them to be bigger. You know, there's things to it. It's genetic, but it's also some environment component that's driving them to be bigger as well. So I think the waters get muddied a little bit when we start thinking about, you know, well, is it just the fact that we're doing this and not this anymore? It's really sort of just kind of a big jumbled mess that I think we're still trying to work through.
Mark, you had a follow-up?
I wanted to follow up on that because at CSU we've done a number of different studies on pulmonary arterial pressure and now bovine congestive heart failure. And so, am I allowed to speculate? Okay, I'm gonna speculate a tad, because we've looked at a whole host of different traits, back to your question, that we would normally measure in our normal performance recording and collect the normal carcass data. And in one of the studies, there was a weak, unfavorable relationship between ribeye area and pulmonary arterial pressure. Hey, it was weak to moderate probably. And that's really which is in contrast to what Justin showed this morning on his paper or in his presentation. So I think that the jury's still out, if you will, on that relationship. But that was one indication. There was another one in one study that we did that said there was and this relates to pulmonary arterial pressure, there was an unfavorable relationship between birth maternal effects, and this is at the genetic level, and pulmonary arterial pressure. Okay, so I've got to think about that, because why would a cow that has the genetics, uterine size or whatever the case is, to produce a bigger birth weight calf have an adverse effect on that calf's genetics for pulmonary arterial pressure at a year of age? And I don't know that I have a good answer for that, but there's some speculation, and I'm speculating, that uterine environment influences maybe lung development or something that influences PAP later on in those animals. And so, I don't want to, that is not definitive, but those are really the only two indications, and ribeye area being a piece of yield grade in retail meat yield. There may be a adverse or an unfavorable relationship with that. We're still looking at that. And I don't know if, Justin, you want to follow. I know you didn't see that in your data set.
I was just going to make a comment about the growth issue, to the point about yield grade. Certainly we've improved quality grade, yield grade's going up, back fat's going up over time. The other big piece that's in the forefront of our mind in this process is the growth. Again, carcass weight is the number one trait in the selection index that's contributing to what when we're selecting for profit. It's carcass weight. Packers know that they want to put as close to 1,000 pounds on each rail as they can. That drives our Charolais type breeds, our dairy cross type breeds. We're pushing those as heavy as they can go. Angus, it's a frame size problem. Angus is not quite at that frame size, and so when we push those as hard as we are pushing the others, that's also probably driving part of the problem. The growth.
So we have another question over here.
Have any of you guys, or you've mentioned about there's a heterosis effect in PAP scoring. Has anybody started quantifying, like F1 weaning weight get 8%? Is there any work being done on, because we got all these black cattle, some of us raise black Simmental cross cattle or black Simmental cattle, just talk about how heterosis plays, or is there some magic in it like there is much of these other traits?
I can comment a little bit, not specifically with heterosis, but just some of the things that we're seeing in the different populations that we're working with as part of this project. The one I talked about today was just strictly beef cattle, and we've got another set of population which is beef on dairy, and so one of the things that we're seeing is, although the beef population that we have, they're primarily Angus influenced, they are crossbred. They're not straight bred cattle. And so, but we're seeing much lower incidence of heart failure in those beef on dairy cattle. So is that a relationship of heterosis? Because I would expect the hybrid vigor of a beef on dairy to be higher than it would be in beef on beef, just due to the relatedness of the species, the different subspecies and everything else. Because the more unrelated they are, the more hybrid vigor you get. So is that a relationship between that it's hybrid vigor, or is it a relationship of is it because growth rates are lower in those beef on dairy animals, but just anecdotally, it's something that we have the data now to look at, but anecdotally we're seeing quite a bit fewer, a much lower incidence of heart failure in those beef on dairy animals.
There's a couple things that make the heterosis thing stand out to me. One of them is that we see a high frequency of risk alleles, especially in British cattle. That would fit with the Angus, Red Angus kind of centrality of this problem. And then we see lower frequencies of that in continental cattle. And it follows a recessive inheritance pattern, so they got to have 2 copies of the risk factor in order to be at risk. It's not a lock for disease, but they're at risk for developing it at that point. And if you can get rid of one of those copies, one of those risk copies, we would expect the risk for heart failure to decrease. Like our negative predictive value on that is that we would expect that 98.5% of cattle without those risk factors, or that risk factor, would not develop heart failure. That's a double negative. I should put that differently. So less than 1.5% of those would go on to become heart failure cases. So the heterosis thing is basically, if you can cross a couple of breeds and get rid of one of those risk copies, you're going to see a benefit to that in terms of the. Now I have to clarify that is not PAP. That's heart failure at the feedlot level. So it's a little bit of a different answer. I'm answering a different question than what you asked, which probably we shouldn't do.
Callie, we got a question?
If we tested our bulls for these, what did you call it, some AARP gene? I know.
We might have to retire some of them on it.
Anyway, is that something that's significant enough that breeders could test their bulls and identify that as something that, because this is a huge problem. I mean,
so the evidence, I just had a graduate student finish up, PhD student, and we ran a prospective cohort study. So what that means is we started with we ran 3 years of cattle, and there's about 3,000 animals, and we characterized them for actually 2 different genetic risk factors. So the ARDC3, we need to come up with a better name for that because that's not flashy at all. We can work on that here, but we did that one, and then NF1A, which was just another one. So the data that came out of that, this is after the discovery, we did our discovery, and now we have a completely fresh set of cattle that we're going to look at. In that population of 2,000 to 3,000 fed cattle, I forget what the exact numbers were, what we found is that across years, accounting for the sex, so we see more steers than heifers, accounting for endpoints like hot carcass weight, stuff like that, what we saw is that animals that had the risk, like 2 copies of that ARRDC3 risk genotype, if they had those 2 copies, they were more than 3 times more likely to actually become a case. So that we can use that number and calculate another number that it's called population attributable fraction, but basically what that number is the amount of heart failures that we could get rid of if we got rid of all the ARDC3 risk. And that number is about 33%. So if we just changed that risk factor, 33% of the heart failures that happened in that yard wouldn't have happened. So what about the other 66%? The other 66% could be other genetic factors. They could be other causes of heart failure because we did not characterize all the cases nearly as carefully. But basically the take home is if you make a calf that doesn't have either one of these risk factors, and we're not even talking about that third one that we found because we haven't put it through the ringer yet. We're still working on that. But there is less than a 1.5% chance that animal will become a heart failure case. The challenge we've run into is that if you have a bull that has risk and you have bulls that don't have risk, there's really we don't have alternative ways to deal with those. They become devalued, they become losers. And it's on a very individual level, which has been an impediment to, I think, adoption, at least one of the impediments.
Dr. Vander Ley, I'm going to piggyback on Jim's question for you. Leoma Wells, Data Genie, LLC. So I believe that the 2 markers you've identified from what I'm hearing are the large effect genes that were in Dr. Heaton's first studies. And that today is what Neogen can pull off of the 100K chip in the panel that was developed. And they report an individual risk as well as a breeding risk. And that's what my clients have been utilizing, right, today to kind of make their decisions. And you're saying that we can have very high confidence that those 2 markers show us a large effect. And we can go forward and look at the breeding risk on the bulls that we're using that 1 to 5 scale, as well as the individual risk of the 1 to 15 times. From what you're seeing in the data, those 2 markers are our large effect and the homozygosity and everything. I guess I'm just asking the confidence level, because from what Justin's presentation, they have a different population. They're kind of looking at more markers. Mm-hmm. But what we can pull off of the 100K today through the Breed Association and Neogen is a great tool to start with.
Yeah, so there's a couple of things about that are probably important to point out. One of them is we do have, at this point, I have a great deal of confidence in the BCHF5 ARDC31. We have intentionally tried to discredit that one ourselves to make sure that we weren't putting anything out there that was bad. And it has stood up well. I would say at this point, looking at the prospective cohort, I actually have less confidence in that particular marker associated with the BCHF2 marker, the NF1A. That one did not hold up very well in our prospective cohort study. We did not see a significant association with feedlot outcomes. And there's a couple couple reasons why that might be happening. That particular marker is not in a protein coding sequence of the DNA. It's actually in a control region, and near as I can tell, we don't really know how those work. So we're not sure what effect that might be having. But the other one has the ARDC3, the BCHF5, different names for the same. One of them's the SNP ID that we've kind of assigned. That one has turned out to be, like it has made it through prospective cohort validation. And I'm pretty confident in that one. The one that's most exciting to me at this point is actually the Jumanji one, because that one is a protective allele. We looked over hundreds of animals that we've genotyped for these three different locations, and we've got one animal out of all those hundreds that had the minor allele of that Jumanji variant that actually had heart failure, which is well within our expected misclassification rate. So we expect to screw up our diagnosis of heart failure etiology at least that often. So Dr. Heaton always says it's early days. It needs to go through the ringer, but it's very exciting that we have a winner maker instead of a loser maker.
Just to add some clarity, Dr. Vander Ley and I had this discussion over there earlier. They've done a great job in their population of being very surgical and going in and tying their markers that they've identified to actual true congestive heart failure cases. So just to clear up our test has been a mass phenotyping effort. So we've phenotyped close to 80,000 animals. You know, if that animal has a heart score 4 or 5, it's in the data set. You know, we're lungs and everything go along with that. Just for clarity, our test at Neogen is an EPD using thousands and thousands of markers. That test is replacing the 2-marker test that Neogen currently sells as part of Identity. So in the next few months, it's going to transition over to our EPD. Our perspective on it is kind of, you know, we used to select for carcass weight and marbling with the old GeneStar tests, those 2-marker tests. You know, they have some utility. We like an EPD that captures tens of thousands of markers across the genome and fits those effects in the model. That's kind of the future of animal breeding, and so that's what we've tried to accomplish with our test, is implementing an EPD that covers a big scale. We're coming at this problem with data. You know, we have the ability to feed in 30,000 more phenotypes in the next 5 years and make this a similar genetic evaluation to something like carcass weight or birth weight. You know, those traits have been well validated. We know that selecting with thousands of markers works, and that's kind of the approach that we've taken. So just a little contrast there for.
Yeah, so I think along those lines, you know, I think our audience is really curious about what are some of the long-term implications of a genetic evaluation for breeding purposes, right? What can we, I think, you know, the question to all of you, what can we speculate is the long-term phenotype that we need to be collecting or be aware of, you know, in the future to possibly build a genetic evaluation, help, you know, do genomically enhanced EPD surrounding a trait of reducing risk of congestive heart failure. I mean, Tim, you're out there collecting a bunch of, you know, PAP phenotypes and heart scores. Are those the 2 phenotypes of interest?
Yeah, so I'm going to back up. Fascinating. So I just, for just a second, we're going to go back and talk on producer level and what I see in the industry, right? And so, yeah, there's a high genetic traits with everything we do. I have experienced one of those long days where a producer used 1 Angus sire and bred 250 head of cattle to that. And so now I'm pap testing those 250 head, and about 225 head into them, we have had 1 or 2 that have had a low pap score. So we're looking at a 99% high. In fact, when they come in and they said, this sire, he just said, turn it out. I mean, it's all high. So that's the world I live in, going, yeah, this is genetic. Did I have a marker for that? No, I did not, which I wish I would have now. But that's the kind of the thing I look into. I'm going back to reflect on that heterosis question because I deal with this on the producer level. We want to take a limousine and we want to take a Simmental, we want to take an Angus, and we want to breed them together for the heterosis, and it works great unless, and I brought this up yesterday as well, and I'm especially seeing it in the limousine right now. One comes in, the limousine pap's fairly decent, and now my Limflex comes in, and it is extremely difficult in some of the circumstances for me to pass a Limflex right now. I'm having a real issue with Sim Angus because what's happening is because we're taking an Angus bull and we're breeding that Angus bull maybe to a Simmental cow or vice versa or whatever, and we're selecting two that are probably carriers that we don't know because we haven't done the testing yet. But when you take two bad ones and put them together, you have a pretty much a realistic, for us, our world, a disaster. Does that make sense? And so that's what I deal with every day, trying to figure out. We used to think, oh yeah, breed black baldies because you're on your way. But if we're breeding black baldies and we pick a bad Hereford on a black cow, what do we got? So I went off on a trend because I just wanted to remind you that heterosis sometimes can work against you in some ways, and that's the circumstance. So for many, many, many years, even as of last month, I've always been accused of having the best Angus herd in the entire world because I will go into a bull test and I will fail high pap the top 5 or 10% of those Angus bulls. And people said, you're just failing them, buying them and raising your old herd that you hide over here. Because I take and I go in and I fail the top 5% of those phenotypic bulls, right? They're the high gainers, they're the high producers, they're the biggest and growthiest ones tend to have the higher pap. So therefore, the better the cattle get, the better the bull gets, the higher the pap goes. And that's been a trend since 1979. So I just wanted to throw that. I don't know if that at all answered your question, but again, I feel at peace now.
Wow, we appreciate that. So I want to pivot just ever so slightly and kind of ask Dr. Enns and Dr. Speidel to think about a roadmap for us as we're envisioning a long-term genetic evaluation. Are the high elevation pap records that Dr. Holt is collecting, are those going to be usable long-term in helping identify a genetic evaluation for congestive heart failure?
I think yes. Okay, now, explain the biology. So the reason or the basis for my reasoning, because if we look at what we've done as far as pap at high elevation and the relationship with pap at moderate elevation, that's pretty high. I mean, it's ultrasound carcass trait level. Okay, and then we take Dr. Speidel's data that looks at pap and heart failure, right? And that's very high as when you get to the feedlot. So I think, we don't have the data to definitively answer that question. Well, we do, we just haven't analyzed it yet, because what we've done is we've taken our research at high elevation and those genetics of those Angus sires we've used in our own research herd that come from a variety of cooperators, and some of those are related to the naive data that Dr. Speidel has generated as two of his ICASA grants, and we'll finally be able to determine the value of a high altitude pap and a bovine congestive heart failure. Because here's the challenge, right? We track we retain ownership on all of our research calves, and they come to a moderate elevation feedlot about 3,000 feet in elevation, right? But the problem was those calves were raised at high elevation, so we don't have a good feel for how that biases what happens in the feedlot. So really what we need is a data set of all naive steers or heifers in a feedlot that have never been at elevation but are related to those at elevation, to actually look at the relationship without the potential for some bias having occurred from birth to weaning at the high elevation. And so we're hopeful that we can answer that question, but just using my logic, I think that it will hold, that we can leverage all of that historical data to hopefully come up with something that helps our selection. I mean, you know what, you're at 498,000 pap observations, so not that all of that would be usable, but yeah, I don't know if you want to follow up, Scott, at all. He may disagree with me.
I don't know that I disagree. I don't know if I am as hopeful as you seem to be. high elevation, we know, and those animals are challenged, we know what happens with that, and we know that's due to just the lack of oxygen and everything else, atmospheric oxygen. So what I'm a little less certain on is, so if we take the high altitude PAP and we translate that to low elevation, I'm not quite sure what we're measuring in terms of that genetic relationship, right? So are we measuring the physiological response of the heart? Are we measuring, whether one's a hyper responder and one's not? Or are we measuring something else, tolerance to, elevation, that type of thing? So, I think PAP actually will differentiate heart score. I don't know where to go with that high elevation PAP at a year of age, and that's something that I think that we are trying to figure out. On a side note, though, we do have some PAPs and high elevation and heart scores on a number of those Rouse cattle, and their heart score incidence rate was much less than what we're seeing from the general low elevation population. So maybe there is something to it. I don't know.
Can I come back to your question on the phenotype?
Sure, real quickly, because we've got 3 questions.
Oh, okay. So I don't know that we know the right phenotype yet, because one of the things that I worry about is that, all right, we've got a PAP EPD, and what happens if we get a bovine congestive heart failure or a heart score EPD? We really need to combine this into some appropriate measure of pulmonary hypertension to not cloud the issue. Kind of like you've got birth weight EPD, calving ease EPD, and really the end is the same, right, of what you want to select for. And we know that the more traits you select on, the slower the progress in any one of the traits. So I don't know that we have a good answer. I think, now in fairness, I think bovine congestive, or the heart scores and PAP are meaningful data. Don't get me wrong. I'm just not sure how any EPD should be expressed as we learn more.
Clearly
that
a lot of
may change.
opportunity. Scott, quickly.
So to go back and answer your question after my whole, diatribe there, I think you need, I think we're going to need all of it. I mean, in terms of, if you just think of what heart score is, and you look at the percent reporting of carcass data, right? It's pretty low. Getting 3%, right? So getting PAP data, costly, expensive, right? We need to keep collecting data. So, ideally, maybe we find something that's related to heart failure, like some other, maybe instead of carcass weight, maybe yearling weight, something that's more, more or densely reported that provides information. Because really, I think in an ideal world, we're not really trying to, we don't want to select for heart score, give our data. We don't want to say, well, we want lower heart scores, but we don't want carcass weight to suffer as well. So are we really, do we really want to look for, do we want a heart score EPD and, try to identify those curve benders, like a birth weight, yearling weight, where we do have heavier carcasses, but lower heart failure predictions.
Yeah, thank you. Wade, I think you have a question next, is that right?
So the question is actually to the animal breeders. And it's along the same lines that Lane was asking, how do we integrate this information into a national cattle evaluation? And Mark, you went to a place about, we don't want a situation where we've got calving ease and birth weight when we're after, a specific trait. So to narrow it down to an ERT, the way I would look at it is the probability of survival due to heart failure, if that's ERT. So how, and then I've learned something I did not know, and I was just talking to Scott after the recess or during the recess about that strong correlation, almost one, between heart score and PAP score. And of course that is under 14 months of age under some stress. So how, knowing all the information, I know it's very cloudy at this point, but you're putting the pieces of the puzzle together, how do you envision, so what's ERT, that's the question, And how do you envision putting the pieces of the puzzle together for national cattle evaluation?
Well, I think it is cloudy, right? So the probability of death, that is something we for sure want to avoid. But I think Dr. Speidel and Dr. Buchanan's data shows that there's also a loss as hearts change in performance, right? Where we're actually seeing in how the animals perform in the feedlot. So I think it goes beyond probability of death, and I'm talking myself into a big cloud because I don't know what the right phenotype's going to be. So if you reduce death loss with an EPD that says probability of survival, are you actually also fixing some of our losses in performance, our loss in feed utilization related to dry matter intake and carcass weight? And I don't know. It's still cloudy.
Say, if we have ERTs for all those things that you just mentioned, the performance traits, I mean, that takes care of itself, right? I mean, you've got the ERTs for those traits, and yeah, I mean, it's not surprising that what you're measuring is related, heart scores related, PAPs related to those performance traits, but we have EPDs for them.
Yeah, but this is a phenotypic relationship, not a genetic, because our genetic correlations show relatively low relationship with heart failure, with that exception of ribeye I talked to earlier.
But to your point about what's the genetic evaluation look like, you know, a two-trait model with PAP and heart failure combined makes a lot of sense to us, you know. We don't have a lot of PAP data because that's just a hard phenotype to get. We need 20 more of Dr. Holt's running around the country to collect that data. You know, in our feed yard, we don't have time to run the catheter down and do all that work. We push cattle through the chute pretty quickly. But one place we can collect lots of data quickly, which is a good thing for genetic evaluation, is in the packing plant. You know, if we can work with more packing plants or more producers to get that data collected, that seems like a pretty fruitful road.
Let's be honest. The first thing you want to do is reduce your mortalities, especially in the feedlot, because you've got all of your costs in those and you get no value back. And thinking ahead to the rest of your program where it's sustainability this afternoon, that's really an industry sustainability issue. If we can get rid of a lot of death loss, it's not only animal welfare, but it's also sustainability in lost resources for our beef industry.
Okay, we're going to probably try to wrap up. We're running just a tad bit long, but I think super fun conversation and discussion today. Gentlemen, thank you for being here. Thank you for all the work that you're doing. I think the overall theme here is there's a lot of work yet to be done, but there's tangible tools available right now that breeders can implement, and there are strategies that we can try to leverage to make feeders' jobs and feeders' lives easier, and hopefully help the industry as a whole. So I'm going to leave it at that, and I think, folks, lunch should be ready. Jackie, do you have a couple announcements? Okay. So, doctors, thank you. Let's give them a round of applause, please.